This is a presentation on fungi outlining the activities covered in Microbiology Lab. The short list of organisms that we'll be covering today would be one of the first lessons in a mycology class. For General Microbiology Lab, we'll take a quick survey of some of the most important yeasts and molds, and we'll be skipping over mushrooms since they're a macroscopic type of organism. Let's first keep in mind what all fungi possess, regardless of which type they are. All fungi are eukaryotic and have nuclei, which would be very different than the bacteria and archaea that we've learned about so far. Also, we can say that their cell walls have chitin, which is a unique cell wall structure to fungi, and it helps to give them strength and durability in their environment. And they have ergosterol present in their cell membranes, which is unique to fungi. Because of that unique characteristic that fungal cell membranes have, it's often the most common antifungal target. So when we talk about antifungal medicines, they almost always are exploiting some sort of way of inhibiting ergosterol when compared to the common sterols of other organisms, like animals and humans, for instance, have cholesterol, and that's one major difference for fungi. And lastly, we'll be looking at the yeasts as unicellular versus multicellular molds. And so we'll start off comparing the two. Here we have a comparison between the various characteristics of yeasts when compared to molds. And so beyond the unicellularity that yeasts have versus multicellular molds, we can see that yeasts will have colonies that are a lot like bacterial colonies growing on Petri plates, although the colonies can be a little bit bigger and appear to have a waxy appearance. Also, we see that under the microscope, the reproduction of yeasts is an asymmetrical event where you'll have a large mother cell that can often be observed that will then be growing small budding daughter cells, and occasionally the daughter cells will not pinch off right away but can start to grow more daughter cells into a long chain. The long chain that sometimes forms with more and more daughter cells can be referred to as pseudohyphae. Hyphae are long thread-like filamentous structures. And occasionally, especially in rapidly growing yeasts, a type of growth called pseudohyphae can be observed. Pseudo meaning false, and then hyphae would normally be something that you would find as a growth structure on molds rather than yeasts. Here under the microscope, we can see a picture of a whole bunch of yeast cells. These are Baker's yeasts called Candida, and some of them stain blue in this picture, and then some of them are clear. And the blue ones have taken in a dye called lactophenolcotton blue, and once they absorb that dye, they're dead. The ones that are still alive have repelled the dye, and so sometimes that's a technique that's used in cellular biology in general to look for live cell counts versus dead cell counts, but the blue dye in this picture just functionally allows us the ability to look at the cells in general, to have some contrast. Molds, here on the right, can be described as having colonies that are velvety or fuzzy in appearance, like this growth that we see pictured down here at the bottom of the slide. And they look like that because of these thread-like hyphae or filaments that grow not only downward into their beneath substrate or environment, but also up into the air so that they can produce spores, little fruiting bodies, structures that are not actually fruit, but are spores that are either asexual or sexual, and then they'll blow through the wind or become airborne and land in other places, and that will help to spread the mold around in its environment. Here are the major characteristics of the yeast Candida albicans as it's outlined point by point here. We have that Candida albicans is the major cause of human yeast infection. There are actually other species of Candida that are less common that can also infect humans, but by far Candida albicans will be the one that is diagnosed day in and day out in various doctors' offices and hospitals across the globe. And not only does Candida albicans cause the common vaginal type of yeast infection, but also it can cause oral candidiasis. And so in the mouth, it's referred to as thrush. And we can say that Candida albicans is sometimes a repeated infection. It can come and go. It can appear more often than people would ever want to consider, but that's because sometimes people end up with chronic candidiasis, recurring candidiasis. There's no limit really to how many times a person could develop a yeast infection. And this is because we know from lecture that there really is no long-term immunity or immunizations, vaccinations that are able to protect us against fungal infections, and including then Candida. In the worst of cases, Candida can get into the blood, and this would be considered or described as a systemic infection. And in these infections, there's a danger for heart involvement called endocarditis. And in these really severe infections, systemic candidiasis can equal a deadly event where in less than a week, a person can go from relatively stable to, grave circumstances. And so we rely very much on a few life-saving antifungal treatments. So these antifungals include things like nystatin, fluconazole, all of the so-called azole drugs, and we'll talk about other ones in a few slides as well. If you refer back to lecture, a more complete list is provided for antifungal drugs. And we can say that the people who suffer the most from candidiasis yeast infections are diabetics, especially. Diabetes is a disease that we know is problematic for sugar regulation and the ability to produce insulin. But unfortunately, diabetes is actually characterized by having less strength immunologically as well. And so Candida, because of a weakened immune system in many diabetic patients, can be a recurring, chronic, ongoing problem. And we can see this also as well in any other people who may be temporarily immunocompromised or fall into a long-term category like HIV/AIDS. AIDS stands for acquired immunodeficiency syndrome. And one of the first signs of AIDS due to viral infection with HIV, human immunodeficiency virus, is the person can develop thrush. Oral thrush can be an early sign of AIDS onset. And oral thrush is also common in infants or young children who are not able to take antibiotics in a pill form. So occasionally people get sick, and a doctor may say, "Oh, take this antibiotic by a pill," but we can't safely feed a pill to a baby or a toddler, and so then they take oral liquid antibiotics. And so when you put a liquid antibiotic into a kid's mouth, they may switch that around and say strawberry flavor tastes good, or whatever it is that that oral antibiotic may have in it. But then the child may end up with a case of oral thrush shortly thereafter. The next morning they could wake up and there can be these yeast colonies growing, and it can cause damage to the mucous membranes, and it can cause a burning sensation and be painful in the oral area or in the throat. And this is because the normal flora that antibiotics will eliminate when treating a respiratory tract infection, most commonly, will then eliminate bacteria that normally will outcompete the yeasts. yeasts and various fungi of any sort are not going to be sensitive to antibiotics that are for treating bacterial infections. And for that matter, there's a separate line here, individuals taking antimicrobial medications, especially women because of the vaginal area. If normal flora, mainly lactobacillus type bacteria, are eliminated inside of the vaginal area, this can then reduce the competition that typically will crowd out Candida albicans from overgrowing. A lot of women could have small amounts of Candida albicans, these yeasts that you see up here, present in the vaginal area, but they're kept under control and no one would know they were present until some kind of an event, like taking antibiotics causes an imbalance in the bacteria. Here you can see a picture. It says here on the exam. This would be if you were taking the lab exam, so keep in mind if you have a lab exam, a slide similar to this will show the mother cell and then a budding daughter cell reproducing asymmetrically. And this is one of the hallmark recognizable ways that you can see that those are not bacteria, but in fact that they are yeasts under the microscope. Here's a yeast that we'd much rather run into and talk about. As opposed to yeast infections, we can talk about a non-infectious Saccharomyces cerevisiae, also known as the baker's yeast or brewer's yeast. And we can break down its name to understand a little bit more about what it does for us. Saccharomyces is referring to sugar, and myces means yeast or fungus, so it is a sugar-loving fungus. And cerevisiae is in reference to the Spanish word for beer, cervezas. So stay thirsty, my friends. The yeasts are there to make us more beer. And this yeast works in a way as to ferment. So whether you're feeding the yeast sugar in the form of making dough, like putting your sugar, water, and flour together along with some yeasts, they'll begin to ferment. Or if you're putting in hops and barley or grapes and you're making wine or beer, then the fermentation process will take that sugar and convert it into CO2, which is a gas that allows the bread to rise, or to put carbon dioxide into the wine or beer, and also the ethanol. And we know that wine and beer have the alcohol, so if you leave it in there bottled up, you can have a bottle of alcohol from the help of the yeasts. But in the case of bread, when you bake it, the ethanol or alcohol will be baked off and evaporate. Now that we've talked about yeasts a fair bit, let's look at molds. One of the first molds that most beginning students learn about is Rhizopus stolonifer. We call that the common cause of black bread mold. So next time you're in your kitchen and some bread or some tortillas has a fuzzy looking cottony white growth with what looks like pepper on top, that could very well be Rhizopus stolonifer. And it's not a pathogenic organism to humans, but let's learn about what some of its characteristics are. If you were to take this growth and put some sticky tape on it called fungi tape, it looks like clear Scotch tape or sticky tape, and then put a sample of it with some dye under the microscope, you would see these various hyphal structures. And these hyphae have specialized parts. The finest little growths of hyphae are thorny looking, and they finger out into these delicate little structures called rhizoids. And rhizoids are specialized for absorbing a lot of nutrients and moisture, as well as for anchoring the organism down in like a plant or a tree into its environment. Here in the lower left, I'll fix a typo. This should say aseptate. Aseptate is a term used to describe hyphae that have no septa or cross walls. Normally, we would see little cross walls that divide the nucleated structures inside of various types of molds. In this case, though, those do not exist. So there are nuclei, although they're not able to be observed in this staining, but there are no cross walls. Also, there are specialized hyphae that grow into the air, generally referred to as aerial hyphae, and these aerial hyphae have even more specialized names like sporangiophore in the instance of Rhizopus stolonifer. Rhizopus stolonifer. So Rhizopus stolonifer's aerial hypha is called a sporangiophore. Other anatomical structures that Rhizopus stolonifer can grow include spore-producing structures. And so on the left here we have asexual spores referred to as sporangiospores. And sporangiospores are located inside of a sac-like structure. This whole structure is called a sporangium, and the actual sporangiospores are numerous, hundreds or thousands of them are located inside of a thin membrane that makes up the sac-like structure. One part of this that's not labeled underneath that looks a little bit like a mushroom or an umbrella, this is called the columella. And then the sporangiophore that we saw in the last slide is the aerial hyphal structure that grows the columella and then eventually gives birth to the sporangiospores and makes this whole sporangium sac-like structure. On the right, we have a sexual type of spore, and this is referred to as a zygospore. Zygo would be in reference to like a zygote. So think of how egg and sperm used to make a zygote, but in this case, we're not talking about a male and female. Molds are, when they have this sort of sexual function, they'll have what's called a plus strain and a minus strain. And this would be somewhat analogous to opposite sex. And the minus strains of hyphae, if they can manage to find a plus strain, they'll sense one another, and at some point they can fuse together and start to fuse nuclei. And when the nuclei fuse, we get what's called a zygospore as a creation. And that is a product then of sex, and it allows the organism to evolve through genetic recombination, as opposed to an asexual event like we see over here on the left, where all of the organisms would be genetically identical. Another extremely common mold found throughout our environment is called Penicillium. Specifically, one of the most common is Penicillium notatum. And notatum is noteworthy for its ability to make penicillin. Penicillin antibiotic was first discovered back in 1928 with Alexander Fleming, who was trying to grow bacteria, but instead he had these contaminating species. The Penicillium grew these fuzzy white hyphal growths with what looks to be green spore structures on top. So when you look under the microscope, you can see the spores and you can see these structures that have a green pigment. And not only are antibiotics, including penicillin and all of its derivatives, highly valuable, but Penicillium also brings us ways to make aged cheese more flavorful. So a little bit goes a long ways. Blue cheese has incredible flavor, and it's imparted by Penicillium's influence. Brie cheese as well. There's even a cheese called Roquefort, and Penicillium roquefortii is a species that helps to make that. One peculiar type of Penicillium is a species called Marneffii, and this was found to be incredibly dangerous as a lung pathogen, and we're lucky that it's not real common. But one memorable story happened when some archaeologists had gone to Egypt, and they had discovered that there was a buried tomb. Sarcophagus was inside of this room that was underground, and flooding had occurred at some point, and all this moisture had gone in there and had grown a green fuzzy Penicillium marneffii, and it was overwhelming. The people who were in that room had breathed in countless thousands or millions of Penicillium spores, And those spores germinated, and they grew in the lungs of some of these people, and it caused a fatal pneumonia in a couple of the people who were in there, of the half a dozen or so that had been part of that expedition. So occasionally we do find that Penicillium can be a lung pathogen, but it's fairly uncommon. The reason it's so dangerous in general, fungal pneumonias that is that the fluid buildup in the lungs is often assumed to be bacterial in its cause, and antibiotics treat bacteria, but they'll do nothing to treat a mold infection. And by the time the doctors realize that what's happening is fungal in its nature, it may be too late to reverse the tides of the rising liquid that can be building up inside of a fungal pneumonia case. Under the microscope, we should specifically take note of the various structures here, starting with the conidiophore. This is the specialized aerial hypha that grows up into the air, and then it branches off into what are structures called phyalids, and these might remind you of like the phalanges of a hand, like a skeleton hand. And then the finger structures extend out as chains of naked spores, asexual spores called conidiospores. And conidiospores can be also referred to as conidia. And these are all genetically identical structures. Penicillium does not have the ability to make sexual spores, so we only observe their conidiospores, and we can say that these also might be memorable as looking like little brushes or brooms that you would sweep a floor with. And so this is how Penicillium looks under the microscope. Also down here are just some drawings to remind you of aseptate hyphae. Aseptate hyphae means that, or I should say, sorry, septate hyphae have cross walls, have septa, and that would be contrasted with Rhizopus, the previous organism we talked about that lacked the septa or cross walls. Lastly, I'd like to point out that fungi are grown on a specialized agar. Most often it's called sab agar, and sab, in reference to Saborod's dextrose agar, is specially formulated to have a low pH. So to be exact, it's about 5.6. It's right in between 5 and 6. And it encourages fungi to grow, which are somewhat tolerant to low pH, below 7, which is neutral. And bacteria, on the other hand, are mostly neutrophiles. And so the majority of bacteria will be inhibited from growing and contaminating what would be a formulation specifically to grow mold. Last fungus that we'll talk about today is Aspergillus niger. Aspergillus includes many species. Niger would be one of the most common and important, and niger refers to its black color that we see down here on this plate of mold colonies. And most strains of Aspergillus niger will have yellow hyphal growths, and then topped with the black spores that we see that make up the black color. Aspergillus is important because it can cause problems of black mold problems growing on building structures. This should not be confused with another mold that can invade buildings and structures called Stachybotrys, and that'll be covered at a different time. But Aspergillus can be a cause of black mold. Also, it can invade the lungs and cause a disease called aspergillosis. And so this lung infection can be severe enough that people who really suffer from it can end up coughing up what are called aspergillomas, or fungal balls. And although they're not contagious and this organism cannot be passed from person to person, people do get this disease commonly from being in environments with overwhelming amounts of mold spores, like when buildings or building materials are damaged due to flooding or excess moisture. And Aspergillus can also, in some instances, be used as a complex aspect of the flavor of soy sauce or soy-based items that have a fermented part to their recipe as they're produced for specific flavors that are aged and fermented. Microscopically, if you were to take a sample of this with some sticky tape, some fungi tape, and put it under the microscope, you would be able to observe the conidiophores. So you can see these specialized aerial hyphae that grow up into the air, and then they terminate into these fruiting bodies. And these are analogous structures to Penicillium, but they're just arranged in a slightly different fashion. So the conidiophore is the aerial part, and then the conidiospores, also collectively referred to as conidia, are still asexual, genetically identical little spores. And these organisms have septate hyphae, so there are little cross walls that divide each of their nucleated cells. And the phyalids, which are not labeled here, I'll write those in. The phyalids are, rather than being in a hand-like formation, like in the previous slide with Penicillium, they extend out radially from the center as finger-like structures that, if you roll the fine focus on the microscope, you'll see them extending out like this. And then the conidiospores grow out from there. This is just one layer of conidiospores in a relatively young growth. And then over here, we can see one that's older and has a lot more conidia or conidiospores that have grown out from the phyalids. Here's another picture of Aspergillus as well. Finally, we'll end today's laboratory activities by making our own fungal tape mounts. So you can take the plates just like you saw pictured in the presentation, and then you take some sticky tape and you'll push that down onto the plates, and then you'll place 1 drop of lactophenol cotton blue, which is a blue dye that will be put onto the slides so as to color the fungi. It also helps then to make the fungi safe for study because it kills them as they soak up the blue dye. And then you'll be placing those under the microscope and looking at the various features that I talked about in the previous slides.